Thank you for joining us for the seminar again today. We'll give everyone back there maybe a minute to settle down, find some seats. I think there are some seats further up front. And then I see a couple in here. So if you're looking for a space to sit, grab those seats. And then it is my pleasure to introduce our speaker for today, Chris Tiessen. He is a master's student in biomedical engineering in the Herzog Lab. And he actually also did his undergraduate degree here from University of Calgary in the HPL in cellular, molecular, and microbial biology with an honors also under Dr. Herzog while studying myofibrils and Titin mechanics. So he just recently started his master's. One of the interesting things about Chris is also that he has recently joined the Calgary Dinos rowing team, which is pretty exciting. The introduction is short and sweet today because you all have probably seen Chris present a few times at seminar now and probably know him pretty well. Chris is amazing because he presents probably every year. Be like Chris. I like it. But with that, I'll let him present his talk titled Titin Interactions in Skeletal Myofibrils. Thank you, Chris.
All right, so can everyone hear me at the back? Okay, so let's get started. So the first thing I want to give is just a little bit of an outline. First, I'm going to give an introduction to sarcomeres and Titin, because I think generally in the Human Performance Lab, our seminars tend to be a little bigger scale, talking about biomechanics. Today, we're going to go down into the molecular or into the subcellular biomechanics of muscle contraction. And we're even going to talk about single protein interactions in muscle. The next thing I'm going to do is talk about a few different interactions of the protein Titin, which is one that we're very interested in the Herzog Lab. I'm going to talk about 3 different things. I originally was going to talk about 5 different things, but I thought it was too much. So I brought it down to 3, and I'm going to give a short story about each of these 3 projects that I've been working on. So the first thing I want to talk about is the organization of skeletal muscle. So skeletal muscle is important in a few different ways. The first is skeletal muscle is a metabolic organ. So this is going to consume energy in kind of a basic sense. It's going to convert chemical energy from ATP into mechanical energy that we use to move our skeleton. Skeletal muscle has a hierarchical structure. So this looks like we have intact muscles. Those muscles are then going to be comprised of fascicles, which are each going to contain muscle fibers. Muscle fiber is often referred to as a myocyte. It is a muscle cell. And then within muscle cells, we have hundreds of these subcellular organelles called myofibrils. And oops, wrong one there. So this is a myofibril. And in our lab, we have the ability to isolate these myofibrils and test these myofibrils. So this is what a myofibril looks like. And you can see this striation pattern is very characteristic of striated muscle. And so just to kind of orient you, these dark bands are called A bands. This is where we find myosin thick filaments. The lighter bands are called I bands. This is where we primarily find actin thin filaments. We can test these myofibrils, and they give us a few different advantages. The first advantage is that they don't have any connective tissue. So when we're testing a myofibril, all of the force production comes from the actual muscle tissue. It comes from the filaments themselves, the actin and the myosin, and as we're going to talk about the Titin. We can measure forces using these cantilevers. These are very small silicon nitride cantilevers that we know how stiff they are. So if we pull on a myofibril, we can measure the force. And then we can move the myofibril on this needle. And so that kind of is our testing setup. Just to give you kind of an idea of what that looks like, I have a little video here. So you can see this myofibril is going to be stretched. And as we stretch that myofibril, we have stress production. In this case, it's just a passive stretch. So what do I mean when I say passive stretch? And I kind of want to be clear about this because passive and active are kind of weird terms when it comes to muscle. So when I talk about passive, that means we have ATP alone present in the solution. I mean, there are other components so that it's isotonic to like what it would be in the body. But we have ATP, which is going to basically make all of the myosin release from actin. So any force that's being produced is not coming from actin and myosin interactions. And then we have the active state where we have ATP and calcium in the solution. So calcium is going to bind to troponin C. We're going to have a movement of tropomyosin, and then we're going to have cross-bridge cycling. So then we can have active force production between actin and myosin. Okay, so moving on, I want to give a bit of an introduction to Titin because that's what the focus of this talk is going to be. Titin is an extremely long protein. It is the largest protein in the body, and it's essentially a gigantic spring. And so it anchors in the Z line of the muscle. So you can see the actin thin filaments here, the myosin thick filament is right here, and Titin stretches from the Z line all the way to the M line. So it stretches the length of the half sarcomere. The I-band region of Titin is particularly interesting because it's extensible. So basically, if we stretch your muscle, so let's say I flex my bicep and my tricep is being passively extended, my I-bands are going to be lengthening, and that's going to be Titin lengthening in those I-bands. And we have a few different domains within Titin that are important. So we have these immunoglobulin domains, we have the N2A region, it's like a signaling region, and we have the PEVK region, which is a big spring. And together, all of these regions basically make Titin act as a nonlinear spring that generates passive force in our muscle. This is important for a few different reasons. The first reason is that Titin basically stabilizes the thick filament. So you can see it inserts into the thick filament. So when we contract our muscles, our thick filaments are basically stabilized on either side by Titin. The other thing that's important is just providing passive force. So our muscles are viscoelastic, which basically means that they can store energy elastically. They can also deform in a viscous manner. But that storage of energy passively, a good portion of it comes from Titin. Couple of interesting things when it comes to myofibril preparations. So in a myofibril preparation, essentially all of the passive force comes from Titin. This is because we have no connective tissue. And when we do experiments where we use trypsin to selectively digest Titin with a very short exposure of trypsin, we essentially reduce passive force to zero. We've also done experiments in our lab where we basically inhibit cross bridges, and then our passive force is identical with and without cross bridges. Essentially, all force comes from Titin in a myofibril. There's been a bunch of experiments that demonstrate that. So this is kind of cool because we can look at myofibril experiments and we can then infer the behavior of Titin because we know that Titin is the only contributor to passive force in a myofibril. So how does Titin work? This is kind of what happens as Titin expands. So the first thing that happens is we have this alignment of all linker segments in Titin. And so what does that mean? Linker segments are basically small disordered segments of proteins that go between an ordered domain of a protein. And so you can think of them as kind of just flopping around in space. So as we stretch a muscle, the first thing that happens is they align. The next thing that happens is that we tend to have some PEVK extension. So the PEVK is a specific region of Titin. Then we can have IG domain unfolding at particularly long length. So these are domains that are able to essentially pop open like a clasp. And then when we shorten, all of these domains can shorten. So hopefully that makes sense. I think this might be a little bit fast, but we do have to kind of get through it in order to talk about some of the other stuff. So the first thing I want to talk about is some observations we've made at the Z line. So the Z line is basically a structure in a sarcomere that is extremely thin, and it's composed of a protein called alpha-actin. And the Z line it basically serves as the anchor point for Titin. So Titin's N-terminus anchors in the Z line. And it also anchors actin filaments. So when sarcomeres are contracting, they're basically pulling on Z lines in adjacent sarcomeres and pulling them together. Now, normally, Z lines are around 100 nanometers in width when we're looking at them. But around a year ago, we started to notice that when we stretched to extreme lengths in myofibrils, we saw basically these Z lines appear to get extremely wide. And this is kind of a weird result. This is a weird result for a couple of different reasons. The first is that normally we think they're 100 nanometers, and the second is in the literature, there's essentially no evidence that the Z disk could ever get this wide. And so we're wondering what's going on with this. So the first thing we did is we quantified Z disk expansion. So we stretched myofibrils from a short sarcomere length of around 2.5 microns. This is right at the beginning of passive force. And then we stretched them to extremely long lengths. So just to kind of orient you, physiological lengths might stop around 3.1, maybe 3.2. And then beyond that is higher than physiological lengths. We start to see extreme Z disk expansion. What was really interesting, though, is after we performed this experiment, I think it's being cut off a little bit, but we did a recovery period, and after the recovery period, the Z disk essentially went back to normal width. I can show you that eventually, but after this recovery period, the Z disk returned to its normal width, so that was kind of interesting, and we were also able to recover passive force after this stretching of the Z disk. So it probably wasn't just damage to the myofibrils, it probably came from actually the Z disk expanding. Now, a couple of interesting things from Titin in regard to the Z disk. So Titin, right before the Z disk, attaches to the protein actin and then essentially complexes with the Z disk. And we know in a myofibril that passive force comes from Titin. And so looking at this, we kind of thought, well, probably Titin is what's responsible for pulling apart the Z disk, because this is a passive stretch and we're seeing the Z disk being pulled apart. So in order to prove this, we performed a digestion of Titin using trypsin. So the way this works is that trypsin is basically a protein, an enzyme that can break down essentially every protein, but Titin is particularly susceptible. So if we do a short exposure to trypsin, we can degrade Titin first before everything else in the myofibril. And we've published some stuff. Venus has published a paper in 2018 showing that basically we can only degrade Titin and everything else remains intact. And when we do that, what's really interesting is that this Z disk expansion, you can see in blue as a function of sarcomere length, is significantly reduced at long lengths. Here you can see the return back to normal post-shortening. So what we can conclude from that is essentially Z-line expansion, this phenomenon that we couldn't really explain, is induced by Titin-produced passive force. So that's kind of interesting. And I want to show one other thing about the Z-line expansion that we noticed that was quite interesting, which is we tried these same experiments in a soleus muscle instead of a psoas muscle. And what we found is that in the soleus muscle, the expansion was reduced to almost zero. And really, to be honest, at this point, I can't explain to you why we observed that. But what I can tell you is that the Z disk in a type 1 soleus muscle has 6 layers, and in a type 2 psoas muscle only has 2 layers. And if you look at the Z disk width here, it's around 0.4 microns, and here it's around, 1.3 microns. It's almost exactly 3 times wider, and we have almost exactly 3 times more Z disks. So I can't tell you that that's exactly why we see this, but it seems to be a type 2 specific phenomenon. So just summarizing what we saw in the Z disks, we observed Z-line expansion. We saw it at extreme lengths in type 2, but not type 1 myofibrils, that's reversed. And this expansion is clearly reversible after a recovery period. The second thing is we confirmed that Titin-related passive force is responsible for Z-line expansion. And at this point, we really can't say why this occurs. It appears to occur only at extreme sarcomere lengths. So we can imagine maybe it's an injury prevention mechanism. It's better to expand your Z disc than it is to tear your sarcomeres, but we really don't know why this is happening. Okay, so that's kind of the first little story that I wanted to share. The next story that I wanted to talk about is about Titin's PEVK. So Titin's PEVK is a very unique segment of Titin, kind of moving from the N terminus of Titin down to the C terminus. And PEVK stands for proline, glutamate, valine, and lysine. And these are amino acids that are essentially the majority of what makes up the PEVK region. The reason it's unique is a couple of different things. The first is that it is intrinsically disordered. So this means that it doesn't actually have any bonds, so any like disulfide bonds in the PEVK region. And essentially all of the stiffness of the PEVK region comes from electrostatic interaction. So basically different charges are attracting each other, and that's what holds the PEVK together. And when we stretch a myofibril, the PEVK lengthens. So over the past couple of years, many of you will remember Armand. Armand and I together kind of developed this model where we can use immunolabels to label on either side of Titin's PEVK. And these labels are very conveniently placed so that if we measure the distance from the Z line to the red label, and then the distance from the Z line to the green label, the difference between those can give us the approximate PEVK length. So basically the distance between our labels can allow us to measure the length of the PEVK. And this is kind of what those labels actually look like. So you can see the red labels here, the green labels here in a myofibril, and these are a little bit closer together than these ones. And so we can take the difference of those widths and estimate the length of the PEVK. This gives us quite a few different things. The first is the PEVK is a spring. So if the PEVK is longer, we know that there's more force on Titin. And we can actually quantify how much force is on Titin using something called a worm-like chain model. So we can use a model that predicts at any given PEVK length, what's the force on the Titin filament. So if the PEVK length is around 200 nanometers, we can predict a force of around 12 piconewtons, same thing at greater lengths. So this allows us to estimate how much force is on the Titin filament, which has a lot of interesting applications. The first one I want to talk about has to do with pH. So this is Dr. Matt Gage, who I've worked with, and he works on the PEVK region, and he thought that basically there were some motifs in the PEVK region that may be sensitive to pH. The reason why this is kind of interesting is that the pH of our muscles changes during exercise. So a couple of factors that do that are the production of carbon dioxide and the production of lactic acid during exercise, basically from anaerobic metabolism, are going to lower the pH of muscles. So if they're lowering, the question is Titin's PEVK pH sensitive at physiological pHs? So after intense exercise, we might expect the pH of your muscle to go from around 7.4 to maybe at maximum 6.1 is the lowest I've seen in the literature measured. So what we did is we took a look at this. So the first thing we tested is that we could use our labeling system at different pHs, and the labeled myofibrils produced similar stress. This is stress at 3.0 microns per sarcomere as a reference point. The labeled ones produce similar stress to the non-labeled ones at each of the pHs we tested. And then we tested the elongation of the PEVK. And so you can kind of see here in this graph, pH of 6.0. We tend to have a significantly longer pH at low and physiological sarcomere lengths. So what that means is at low pH, we tend to have a more compliant PEVK region. So then we calculated the elastic modulus of Titin's PEVK at these different pHs. And what we found is that at a pH of 6.5 and 6.0, we had a significantly reduced elastic modulus. So reduced elastic modulus, what this basically means is that the pH is making the PEVK more compliant. So that's exactly in line with what we thought. So what we found is that Titin's PEVK is pH sensitive at extreme yet physiological pH in intact myofibrils. And we measured the changes in elastic modulus and found that they can be different up to like a twofold difference at a pH of 6.0 compared to 7.0. So this is kind of interesting. What this suggests is that after intense exercise, Titin is becoming more compliant due to changes in pH. And these pH changes are likely due to CO2 and lactic acid production, which other people have shown basically happen whenever you're doing anaerobic exercise. Okay, the next thing I want to talk about is also to do with Titin's PEVK, but you'll notice there's something a little bit different here in this sarcomere, which is that we think in our lab that Titin can bind to actin, and this is responsible for something called residual force enhancement. So I know that most of you, as part of the HPL, are probably familiar with residual force enhancement and Titin, but I'm just going to go over it anyways for anyone who's not familiar with this. So residual force enhancement is an innate property of muscle. Basically, in black, we have an isometric reference contraction. So we have a muscle that has been activated at a certain length, and the length kept constant, and the force is going to increase up to a maximum, and that's going to be our maximal isometric reference contraction. If we take a muscle at a shorter length and then activate and then stretch that muscle to the same final length, it produces an increase in force. And this increase in force persists, and it's called residual force enhancement. For a lot of reasons and a lot of evidence that I can talk about if we need to, but I'm probably not going to go into right now, we think that Titin is probably responsible for residual force enhancement. And the mechanism that we believe is occurring is that when you activate a muscle, we have Titin-actin binding, which is occurring at the N2A region of Titin, which is then conformationally restricting this region of Titin, leading to the overextension of distal Titin, which is causing an increase in passive force. The reason why we kind of believe this to be the case is that residual force enhancement, it appears to be a passive element that's responsible for residual force enhancement, so it's not associated with increased ATP consumption. Other kind of pieces of information is that if you do residual force enhancement and you stretch to longer lengths, you get more residual force enhancement, which is consistent with the idea of a spring stretching. And so based on this idea, we can make a few predictions. And notably, since we can measure Titin's PEVK, if we think the binding site is here, during this residual force enhancement, we would expect Titin's PEVK to be longer in this state than it would be in this state. So if we had an isometric reference contraction, we expect the PEVK to be shorter than in a residual force enhancement state because we have this conformational restriction that's causing this overextension of distal Titin. So some of you may remember some of this work I did with Armand. Since then, I've done a bit more work on this. Basically, what we did is we measured the length of Titin's PEVK in an isometric and residual force enhancement condition. So this first graph shows that we can achieve residual force enhancement in these labeled myofibrils. So the labels don't affect residual force enhancement. This is the location of those labels. So what we did indeed find is that the green label was significantly farther, and so the PEVK was significantly longer, which is right here. The average PEVK length in an isometric state was around 205 nanometers, and in a residual force enhancement state was 257 nanometers. We used the worm-like chain model, as I was previously describing, to basically predict how much of residual force enhancement this was responsible for, and this accounted for approximately 75% of residual force enhancement, which to us was very exciting because we previously shown that calcium binding to Titin might account for around 20% of residual force enhancement. And so if you add 20% and 75%, this kind of works out almost perfectly to explain residual force enhancement. So basically, to summarize that, we showed that Titin's PEVK is significantly longer during residual force enhancement, and we conclude that this is likely indicative of Titin-actin binding. But what I'm going to note is that we don't know that for sure, because it could be that this region of Titin is getting stiffer, and so that gives the effect of Titin-actin binding. We have some reasons to believe that's not the case, but really, this is an indirect result. We are measuring the length of an element of Titin. We're not directly showing that there's actually Titin-actin binding. So this kind of leads me to our big question, which is what if we could image Titin-actin binding directly? And this is what I'm going to talk about kind of for the rest of the talk. I don't have any results on this yet, but this is some really cool work that I kind of want to share. So taking us even farther away from, I guess, what would be normally considered human performance, let's talk about cryo-electron tomography. So cryo-electron tomography is a microscopy technique that allows us to basically get resolution of individual molecules inside of a tissue. So this right here is the Titin Cryos microscope that is, I don't know, maybe 15 feet tall at UBC that we've used for this. And the way this works is that we load our sample onto this microscope, and then our sample is going to tilt through a variety of different planes. Then through each of these planes, we're going to take an electron microscopy image of our sample. And so we get an image from all different angles of our sample that we can then use to essentially make a 3D reconstruction known as a tomogram. And what's really interesting is that this process, because we're essentially averaging a bunch of different images, really greatly enhances resolution. And so we can get down to a resolution of around 4 angstroms, which is enough to see individual amino acids using this technique. So what I've been working on in the past little while is how we can do this on myofibrils. And the real difficulty is that cryo-electron tomography has previously been done on all sorts of different cells, all sorts of viruses, bacteria, but there's never been a mechanical manipulation of those cells. And this is something that's really critical if we want to look at titin-actin binding, because we need to be able to put a myofibril in a residual force enhancement state. And so what we were able to do is make our myofibril apparatus compatible with these grids that are used for cryo-electron tomography. So here you can see a myofibril held right above that grid. There's the needle and the cantilevers, and here's the grid. And then we can place down that myofibril and remove the grid, and the myofibril stays on the grid. And so this is the same myofibril that I put down here, just imaged after we remove the grid, then put it back on. So we seem to be able to do that. And then what we do is we essentially plunge that grid into a bath of liquid ethane, which can freeze it extremely quickly, which results in the production of vitreous ice. So vitreous ice, it basically has no crystalline structure. And what that means is that the protein structures are preserved. And so you can kind of see this process in the video here. It's kind of cool. It makes me feel very scientific when I do this. So basically it's plunged. You can see right there, that's the liquid ethane bath. The grid is going to be right there. And once it's plunged, it needs to stay frozen until it's put on the microscope. So this is what the microscope looks like. I think you can see Amy, the technician here. So that kind of gives you an idea of scale. It's absolutely enormous. And then we're able to see myofibrils. So this is just a collection of myofibrils. The first images I did were not of mechanically stretched myofibrils. They were just like a suspension, so they're kind of everywhere. This is like a low resolution image. And then this is a higher resolution image. This is a single image, so it's not a tomogram. And you can see this is 20 nanometers. So these are actin filaments in the eye band. And so we get unbelievable resolution doing this type of system. So I haven't yet been able to image the mechanically manipulated ones yet, but that's kind of what's coming. So just to kind of give a summary of this, our custom built system enables imaging of myofibrils, and we can correlate those to mechanical parameters at a sub-nanometer resolution. So the reason why this is so important is we can actually look at these other things that I've talked about. So things like the Z disc expanding, things like the PVK being pH sensitive, things like titin-actin binding, and we can actually go in and see like what are the proteins actually doing to confirm if what we think is happening is actually happening or not. So we have the potential for revealing the molecular structure of the sarcomere in different contractile states. So the last thing I want to do is just give a little bit of a summary. I hope I didn't go too quickly here. I kind of had maybe too many things to say, but the first thing is that we showed the Z-line expansion that should say type 2, in type 2 myofibrils due to titin-produced passive force, kind of a novel result that hasn't been described before. The next thing is we show that the PVK is pH sensitive and it can increase its compliance up to twofold at physiological pH. We also show that the PVK is significantly longer in residual force enhancement compared to isometric contractions, which gives strong evidence that Titin-actin binding is responsible for residual force enhancement. And then kind of the future is that we're using cryo-electron tomography to reveal Titin-actin interactions that might be associated with mechanical changes and what's explaining those mechanical changes at the molecular level. And that's pretty much all I plan to talk about. So if there's any questions, I'm happy to answer them.
With that, the floor is open for questions, and who would like to start us off? Oh, Steph.
For your Z disc widening, what do you think the implications are for that? Like, are there assumptions in terms of how we think about muscle or like how we calculate things, where that will affect it? Like these assumptions that we've been making in order to, let's say, calculate sarcomere lengths or something. Like, what changes because of that?
Yeah, so the first thing is that it appears here, I'll go to this slide here. So what we also did is we did some electron microscopy images in fibers. And those ones, even when stretched to the extreme lengths, it was really rare to see that Z disc expansion. So it appears to not happen as much in fibers. That's probably because of Desmond reinforcing the Z disc in a fiber, but not in a myofibril. So it might be kind of a myofibril specific finding that happens because you have an isolated myofibril preparation, which makes the Z disc weaker. In which case the implications would be that our measurements of Titin length might not be all that correct at long sarcomere lengths, because if the Z disc is expanding, presumably the Titin insertion into the Z disc is also moving away from where we think the Z disc is going to be, if that makes sense.
So just kind of a follow-up. So that protein that you said that was a Desmond, why is it not in the myofibrils, but it's in the?
So the Desmond links adjacent Z discs. So it would, link this Z disc to this Z disc. Yeah, so it probably is in the myofibrils. It's just not linking to an adjacent Z disc. So it's just kind of sitting there. Whereas in a fiber, it's still probably intact when it moves Z discs.
Right.
Thanks, Chris. I love those images. I think I'm gonna sneak a piece of phone into your pocket next time you go. Don't take this the wrong way with this question. It's an honest question. If you observe that Z disc expands when you place the myofibril under tension, passively, and you know that all of the passive force production in the myofibril is generated by Titin, why do you have to do an experiment to show that it's Titin that's stretching the Z disc? What else could it be?
Well, the real answer is it's kind of redundant, but I wanted to learn how to use trypsin in myofibrils. So I wanted to learn that protocol and kind of a why not thing. We kind of already knew it was Titin. But the other thing too, is that we had a little bit of confusion because in myofibrils, we don't always have exactly the same stress in different myofibrils, but we still see Z disc expansion in all of them. And so one of the things I was able to do with those trypsin experiments is do a partial digestion. And then if you normalize to stress, the amount of Titin predicts the amount of Z disc expansion as well. So I didn't show that here, if that makes sense.
Yeah.
Micah and then Steph.
Thanks, Chris. I'm curious about your, I guess, the middle portion of the results that you were sharing about compliance of Titin, I believe. So you said that when the pH kind of is reduced, you would expect to see Titin that's a little bit more compliant. So I'm wondering, like, A, what are the kind of injury-related implications for that? due to exercise and aerobic exercise where we would see higher kind of, or lower pH levels. And then the second part to that is like comparing a highly trained individual versus maybe a recreational individual. If we're doing the same like level of task of exercise, would you see significant differences in fun?
Okay, so for the first part of that question, I'm not really sure when it comes to injury prevention, because it's not all that clear to me if more compliant Titin is better or worse. Because if you have more compliant Titin, maybe your muscles are passively more compliant, but I don't know if that makes you more or less likely to be injured. And I could probably come up with reasons as to why both might be true. I mean, if it maybe more compliant means you're more likely to, overstretch, and less compliant maybe is better in other ways. So I'm not really sure. In terms of training, there's a couple of different things. So, I mean, somebody who is aerobically trained would probably be less likely at a given level of activity to be doing an anaerobic response. Because they had more aerobic capacity, so they might have less changes to pH in their muscle. Having said that, if you're extremely well trained, maybe you are used to working anaerobically, you can really push yourself. Maybe you can actually get down to lower pH levels when you're actually pushing yourself because your lactate threshold is higher. So then we might see more compliance, if that makes sense.
So kind of, you might have sort of answered the last one, but so you said that in the literature it goes down to 6.1.
Yeah.
How often does that occur, or like what is the typical range of those?
Yeah, so the literature for pH in muscles, I've seen 3 different papers on it. 6.1 was one, is a paper measured from a long time ago. It's like 1980. I forgot the author. But the other 2, they tend to measure around 6.5 as the lowest pH that muscle hits. And it tends to have to be extremely intense for that to occur. So we're talking like anaerobic activity until failure. Like, I don't know, you're running like an 800 meters. At the end of the 800 meters, you collapse. Your muscles are around 6.5 pH type of thing.
So is that in the cells, or is that like where in the muscle are these measured?
I think those are measurements that are just like in the muscle, and so they're not entirely sure where that is. Like it's just like a pH probe that is stuck into the muscle. We would assume that the intracellular pH, like inside the cell, is going down because this is going to change because of CO2, which can basically dissolve across membranes, and because of lactate production, which would presumably happen in the cytoplasm of the cell, glycolysis. So I would imagine it would be the intracellular pH. Yeah.
And then in terms of like the functional implications of the reduced passive force, like what do you think the implications are for like whole muscle?
I think it's probably a little bit less than it seems. The first is because Titin is only responsible for around 50% of the passive force in an intact muscle, because the rest comes from extracellular matrix and collagen. I think the other thing to keep in mind is that this is only the PEVK segment of Titin. And so other segments of Titin don't necessarily have the same response to pH. And I don't know what their response would be. The other thing is that Titin's PEVK is a nonlinear spring that generally has a roughly exponential curve as a function of length. And so this means that an increase in compliance doesn't necessarily equal that much of an increase in length, if that makes sense, because it's an exponential curve. Yeah.
Okay, we had an online question and then Brett.
Okay, let me read it up here. I was wondering how you could confirm the hypothesis on a larger level. For example, you hypothesize the expansions of the Z-band for injury prevention. How might one go about confirming this is in a non-destructive way in a moving being?
Well, I can say this. So, study from Sweden in 1989 looked at damage to the Z disc on after eccentric activity. And it looks very, very similar to this. And it also looks like expansion. This is a phenomenon that was then called Z-line streaming, which has been observed in muscle in response to eccentric training. I cannot confirm that that's the same phenomenon that I'm seeing, because what I see in myofibrils is reversible, and there's no evidence that that evidence from an intact muscle after eccentric training is reversible. But it's possible that those two things are linked because they produce kind of similar looking images of the Z.
Hey, Brent.
This is kind of a play off of Jess's question about pH. Like, there's no doubt that the pH decreases with exercise. But how are you convinced that it's lactic acid? Like, I don't know if lactic acid can even survive in a pH above 6. I'm not even sure the formation of lactate is going to consume a hydrogen ion. That's presumably going to work to keep the pH higher. So, like, how do we confirm that it's lactic acid that is responsible for driving the pH down, and these other observations that you see with muscle or Titin compliance?
So I'm not 100% sure, because this is not something that I would do. But from the studies that I've looked at, who look at how pH changes in muscle, they generally basically attribute lactate production to a certain pH. So they'll measure like a concentration of lactate and predict the contribution to pH that that would have. And then the amount of CO2, which then undergoes a reaction to produce carbonic acid. And I believe that those two are kind of the main contributors, at least based on the studies that I know. I'm not exactly sure how they know, like, how much is from lactate versus how much is from CO2. Yeah, not exactly sure. I know MRI spectroscopy can measure lactate concentration, so that would be really cool to do.
I have a quick question for you, Chris. You mentioned that the type 2 compared to type 1 is what has the Titin produced passive force that causes the Z-line expansion. Is that because the type 1 don't have as many Z-lines? Is that correct? Or am I mixing two things?
They both have one Z-line.
Right.
The Z-line is a scaffold. Let me go back to this slide. So the Z-line is a scaffold that is basically made of alpha actinin that is kind of in this lattice arrangement. And they have different numbers of layers in their lattice. So in a type 1 muscle, they have 6 layers of alpha actinin. And the interesting thing is, if you imagine force being transmitted to the Z disc, it comes down the actin filament, because Titin binds to actin just before it enters the Z disc. And then it would have, 6 pathways in parallel to be transmitted. It's not, this is in 3D, but it's a 2D representation. Whereas in a type 2 muscle, it would only have 2 pathways. And so if we think about that, just like force sharing in parallel, each of these would only need to support 1/3 the force of this. And so if they were similar stiffness, this could explain our results. Now, this is just kind of a speculative hypothesis that might explain what we're seeing. If that makes sense.
Is there any way to test that hypothesis?
Yeah, we could use cryo-electron tomography.
Okay,
Basically,
that's
yeah.
the thing you're trying to do.
That's what I'm trying to do. Yep.
Glad I got it. Other questions for Chris? Walter and then Venus.
Yeah, you just go first.
Okay, we know Walter's gonna ask one question.
No.
I'm thinking about the effect of pH on Titin. And so change in pH is usually happening in active muscle state, not in the passive. So do you think the Titin actin interactions could be also pH dependent, and it could affect like how much eccentric force you get from Titin and residual force enhancement?
So I think the interaction itself is probably not pH dependent. And that's just because when I measured the PBK, I also measured the proximal immunoglobulin domains, and they had no change in their length with pH. So it doesn't look like pH is changing their conformation. However, I would expect if PVK is becoming more compliant, maybe we have less residual force enhancement because the Titin actin binding site would be proximal to the PVK region. And so if we had a more compliant PVK, then if at a longer length would produce relatively less force than if the pH was higher.
But the active force is less as well.
I guess that's
because
true.
of the
Yeah.
pH.
Yeah.
So do you think, how do you see them working together, the passive force becoming more compliant and the active force becoming also reduced?
So the passive force probably doesn't play as much of a role as the active force being reduced because of pH effects on cross bridges. The reason why is just like at most sarcomere lengths in vivo, we would have very little passive force. And even when we do a long stretch for residual force enhancement or something like that, our passive force component is usually like, I don't know, something around 30–40% of our total force. So effects on the active force are probably relatively larger than the effects on the passive force. If that makes sense.
Just a simple question at the beginning there. You know, when you mentioned the elongation of Titin initially, you mentioned this sequential elongation that I guess was suggested by Grancy at one point. I know that you and Armand have indications that it's actually not that way, that it's more a simultaneous expansion of all elements, proximal, distal, IG, and PVK. And I wondered why you chose to go with one argument where you and Armand have found that it's probably not correct.
Yeah, I can go to this slide that kind of shows that. So that figure that shows the sequential elongation, I put in because that's kind of the conventional hypothesis, just to explain how it works. And we haven't published that segmental elongation yet. So I didn't put that in. But yeah, that's absolutely right. So for example, we can measure the distal immunoglobulin domains, and at a sarcomere length of as early as 2.5, we start to see lengths that are above their fully folded contour length. So we're seeing unfolding as early as like 3 microns per sarcomere, which is essentially not sequential at all, and everything seems to be quite simultaneous. So I guess I just put that in there because it was.
But you believe essentially the results that you and Armand had, that probably that sequential thinking is probably not quite correct.
Yeah, I mean, it still does seem to be somewhat sequential in that there seems to be a PEVK dominated region that is at the beginning of extension, and that the PVK reaches its end of extension around 3.7 microns per sarcomere. And then after that, it's 100% immunoglobulin domain. So in that sense, it's sequential. But during that PVK extension, it seems that the immunoglobulin domains are unfolding. And we see it every time with myofibrils. Like I'll stretch a myofibril to like 2.8, 3.0 microns per sarcomere. And when I shorten, I can see the immunoglobulin domain refolding. So it's really clear. Yeah.
And the other question that I had is, you know, when you talked about the PVK and the pH experiments and also the Z-band expansion, so I think they were all done passively. So would you assume that if you activated the myofibrils, you would have similar results or different results? Or
yeah, so talking about the Z-disc expansion, I don't have a figure of this on this presentation, but it's really interesting because the maximum force, I think maybe I can show it here, maximum force we get in a passive state is around 250 nanonewtons per micron squared. That is very similar to maximal active force, and yet we do not see any Z-disc expansion in the active state. It's unclear why that is right now. One thing is that alpha-actinin appears to change conformation in the presence of calcium. So that might be, that change in conformation might be strengthening the Z-disc. So that might be an effect of active conditions on the Z-band. In terms of the PVK and pH, I'm not sure if activation would change very much. Other people have tended to say that calcium makes the PBK stiffer. So calcium is making the PBK stiffer and pH is making it less stiff. Maybe overall we have a stiffer PBK at low pH in an active state. However, Daria did those experiments and he didn't see any stiffer PBK in the presence of calcium. So it might be very similar. In the active state, we would expect Titin actin binding. So then in an eccentric state, I would expect a longer PBK. So yeah, that's pretty much what I'd expect.
Not necessarily that the pH and the pH results that we have in the passive would be conceptually different for the PBK modulus or elasticity.
Probably not. Yeah, probably not, because at least the results from Matt Gage suggest that it has to do with poly-E motifs, which are collections of glutamates that are probably not changing in the presence of calcium, based on some other work that Matt Gage has done with calcium and the poly-E regions.
I was just thinking, when you have a low pH and you're presumably tired, as you said, at the end of an 800-meter run, and let's assume that, on the sarcomere level, indeed you would have to tighten that's a little bit more compliant. And maybe that additional compliance when your activation of the muscle might not work properly anymore because you're really tired and you might get micro tears, maybe that additional compliance will prevent more serious injury compared to if it was stiff, right?
Yeah.
Something like that.
Maybe, yeah.
Maybe, but both these observations, also Zeeland and the PBK, more compliant. And at the end of the day, I guess everybody was asking, is there maybe a functional relevance? And of course, there's always the possibility that it might not be.
Yeah.
But it's just something that happens biologically, that from an evolutionary point of view, from a functional point of view of the muscle, you might not really mean.
Yeah. What I will say is that the PBK being sensitive to pH is based on the poly-E motifs in the PBK. And those are conserved among species. So if you look at the Titin in a frog, and the Titin in a rabbit, and the Titin in a human, they all have those poly-E motifs. And they don't appear to have a function that we know of except being sensitive to pH and basically conferring stiffness to Titin. So there's probably something relevant about the fact that Titin is sensitive to pH. I would argue the opposite on the Z-disc. I would say that the Z-disc result is probably an artifact of a myofibril preparation because it's really only at extreme lengths.
And of course, the poly-E motifs, Hank and Siegfried Nabei, have argued at one point that they are the ones that get stiffer with the calcium, in the presence of calcium. So therefore you have that dual effect potentially on the same elements where you modulate stiffness somehow of Titin, either stiffer or less stiff depending on how tired you are and how much you activate them. Which it's an interesting thing that you have a spring that we know changes from species to species, that might also be instantaneously modulated to a certain degree by just what happens in the environment of the sarcomere.
Yeah. Any other questions for Chris? Well, if not, then let's thank him for his talk.